Hello, everyone. Today I will be presenting an article called New Codons for Efficient Production of Unnatural Proteins in a Semisynthetic Organism. This article is published in Nature Chemical Biology, and where the experiments are done is the Scripps Research Institute, which is located in San Diego, the southernmost part of California, and it is considered one of the best biotechnology companies in the United States, so that makes it one of the best in the world. So this article is written by Rosenberg Labs and his PhD students, and it is about a revolutionary method for changing the whole central dogma process by adding unnatural base pairs and non-canonical amino acids into the central dogma. So I will start giving a small introduction from the very beginning of the biology we know. So I want to start from nucleic acids. As we all know, nucleic acids contains three main parts, which is the base and the sugar group and the phosphates. The base is where it gets its identity, and it's mostly based on nitrogen, as you can see here. And the purines and pyrimidines are have different structures. The purines have a ring, like 5 carbon ring and 2 nitrogens, and this one has a pyrimidine ring. And the main difference between ribose and deoxyribose is that ribose has an additional oxygen in the second carbon, which makes it way more reactive than its counterpart deoxyribose. So again, like a little more introduction, I'm going to talk about the terminology of how they're called. Once without the phosphate group, the sugar and one of the bases is called nucleoside, and when it gets the phosphate, it's called nucleotide. So as I mentioned before, purines are here and pyrimidines here, and they make hydrogen bonds. But in this article, I will present something that is slightly different than this. So I want to talk about a bit timeline of the unnatural base pairs. They're still a recent research topic in the field, and I want to start giving an example of Hirao's unnatural base pairs. These UBPs, as the abbreviation, were first developed way before in 20th century, but most efficient were developed in the beginning of the 21st century by Hirao, which is a Japanese scientist that developed a UBP that is replicable by the DNA polymerase. But I'm going to be talking about this specific UBPs that are published in 2016. This one published in 2016, and this is 2017. They replaced D56 base with DTPT3 because it is slightly more efficient than this one. So about these bases, as you see here, they're slightly different than the natural adenine and thymine bases because there's no hydrogen bond made by the structure. They use hydrophobic bonds because you can see there's no oxygen or nitrogen here. They're only made of carbons, and this is sulfur, and sulfur is also quite hydrophobic. So they recognize each other by using hydrophobic forces rather than hydrogen bonds. So they make hydrophobic bonds, and these ones are replicable by the DNA polymerase, which means that they can be implemented, multiplied by the PCR, and also multiply inside the cell without intervention. And they have high fidelity. The normal DNA is almost 99.9% fidelity, but these ones still has high fidelity of 98, 99 for most of the cases, and that's even inside the cell. And one of the best properties of these UBPs is that they can be transcribed into mRNA and tRNA, so they can join the complete central dogma cycle, not just the DNA. I will present the strain which they used, and also this strain is the one that I presented for my first year in club presentation. I want to retell everything else. This specifically, this strain contains a triphosphate and nucleotide triphosphate transporter that is capable of transporting nucleotides into the cell. So as we know, the bacteria can't get the ATP out of its cell. So with this transporter, we can give bacteria the base pairs that we want from outside. So during the replication and transcription, we will be able to give them DNAM and DTPT3 UBPs inside. And one of the other properties of this is amino acylation of tRNA with non-canonical amino acids. Non-canonical canonical amino acids are just amino acids which are not found in nature, they are synthetic. So I will get that point. But this one means that they can be charged by amino acids and they can join the translation process. So that also makes it really useful for translation of proteins. And it allows in vivo transcription and translation of these genes, so which is basically the point of why they choose this one. So I will present non-canonical amino acids. They're just like normal amino acids, but the main difference is they're not synthesized in the cell. They're mostly unnatural, and that's why sometimes they're also referred as unnatural amino acids. And this is abbreviation. They have quite a lot of advantages in biology. One of the first is they have uncommon bioorthogonal chemical groups that are not found in nature, like this azide group, which is not found in nature and could be used in very different processes. For that reason, they have diverse functionality and applicability in biology and nanotechnology. So by means of creating a drug delivery system that has an affinity to like a bioorthogonal chemical group, such as azide, we can incorporate an amino acid into a protein, and we can have like a drug targeting system with using these sort of technologies. And for nanotechnology, it's like a bridge between the nanotechnological products and the proteins that has these sort of amino acids, because usually the normal amino acids don't really interact with the nanotechnological products and other chemical groups. So that's why they're really important in the future, maybe. And other advantages is contains that they don't take almost any metabolic fat base. Many amino acids take part in metabolic fat base, such as glycine joins the citric acid cycle, but most of these things are not touched by any enzymes, so it's really easy to maintain their presence in the cell. And they have a large variety of choices. I mean, you can synthesize a lot of non-canonical amino acids because the carbon already has a huge variety, and we can manipulate this for future uses. So just want to give an outline. In normal systems, the mRNA reads the anticodon, finds the codon, the triplets, and they recognize each other, which gets the correct amino acid, and then the translation occurs. And after the translation, we have proteins with mostly 20 canonical amino acids. But in the system, they wanted to develop a system which they can add unnatural base pairs, UBPs, into structure so that they can expand the codons. Additionally to these 20 canonical amino acids, they also can add non-canonical amino acids in the structure without disturbing the natural one. So overall, this is like the total integration to the central dogma. In replication, by adding these UBPs, and transcription, same. And for translation, also adding non-canonical amino acids into the protein structure. So in this article, they claim that they can they analyze multiple unnatural codons and anticodons. So there are experiments made on this. I will show that. And they identify 9 unnatural codons that can produce unnatural protein with nearly complete incorporation of non-canonical amino acids with diverse variety of them. And at least 3 of them can simultaneously incorporate 3 amino acids in the structure. So that makes this being with an organism with first 67 functioning codons. So that's like a novel thing that's never been done before. It's always considered as 61. So I will dive into results. So in this experiment, they use these specific 3 non-canonical amino acids, but they mostly use AZK and AZF. They didn't use this much. So what they did is use a superfolder fluorescent protein that is a special protein that is mostly used in similar studies like this, that tolerates some amino acids in specific places, like the 151st codon. So they want to change the codon at this one so that they don't disturb the structure of the protein. And again, I said that. And they also add like a tRNA. This prilosine is for the control. This is going to be replaced. So they use the tRNA so that in translation, it can make a tRNA and get amino acylated by one of these non-canonical amino acids and incorporated in the protein structure. So first, I want to mention a technique which is commonly used in this article called Spec. So this is one of our amino acids that we're going to use, and this is like another compound that has huge affinity with this amino acid, and they bind and create like a fluorescent structure, so that if the amino acid is incorporated in the structure, when they send a specific wavelength light, and if that glows, that they will see that this specific amino acid is binded to the structure. And the reaction is almost 100% efficient, but it can give very efficient results as well. So here we see about 16 or even more experiments done on using the codons. So the first one is without anti-codons, so it doesn't code any protein with the unnatural base, the amino acids, the unnatural amino acid. And the second one is uses complete natural system without the X and Y. And let me clarify, X is the and Y is the DTP3 UBPs. And in this structure, we see that they are completely natural, so it's really efficient without AZK in the medium. Is almost not, there's no fluorescence, and with AZK in the medium, there's huge amount of fluorescence. And some of these codons are not very efficient, as you see. Without it, there's even more fluorescence. So this is not a reliable codon for the experiment. So what they did is they choose the ones that is similar to this one, which means a low fluorescence in without AZK and high fluorescence with AZK. So they choose probably this and these sort of things. And they made a chart and they identified 7 of these codons. So this allows them to find out which one is the most efficient one. And they further analyze the codons. As you see, it's even more clear in this chart. Some of these are really nice, that almost like this one. And they measured the fluorescence levels by changing the codons. And one of the main differences between the variety here in the fluorescence is because these UBPs use hydrophobic forces, which is very different from hydrogen bonding. So simply, they have quite different chemical properties. So it's important that whether the X is on the middle or the other base pairs next to them. So that's why they did experiment like this. And they also analyze it with mass spectrometry to make sure that the protein is working fine. So once they did that, they switched to adding decoding 2 unnatural codons, because first one is easy and they already did it in 3 years. So they did a similar structure, but this time they used 190 and 200 part of the protein, which is also has similar effect like the 151st. they added 2 tRNAs. This is again a control group, so these are going to be replaced. So in the actual experiment, they replaced prilosine with AZK, and AZF is AZF. So when they did this experiment, you see that in the Western blot, these ones look heavier, simply because they've been incorporated by amino acids and also applied by Spec reaction that makes the protein heavier. So, but they use also another amino acid which also includes a special reaction called QAAC, which is pretty much like the Spec reaction. This also adds an alkali to an azide group, and when you send like a light, it emits light in a different wavelength, so you can identify the fluorescence. So the green fluorescence came from the protein. You see there are like 3 letters. So this means one in the bottom that there's no QAAC and Spec done, so it's still not really heavy. But once we do Spec, it just gets a bit heavier, and once we add the QAAC, it gets even heavier. So that means that something gets incorporated in the structure of the protein. This is unnatural, because we know these substances, alkalines, react rapidly with the unnatural proteins that we use in this structure. So once we send this different wavelength, we see that there's a fluorescence happening here, but not in the one without this reaction. So once these images overlap, they look like this in the Western blots, which looks really nice, I would say. So this one is a more general view of this. They did it both, and they also did it separately to make sure like how the expression works. So you can check the results in more detail in the article. So they did a mass spectrometry, and they realized that almost 92% of the isolated protein contain both of the non-canonical amino acid in the structure, and almost 8% of the proteins had missing one of them. So that's mostly caused because X to D mutation, which is X is DNAM to the thymine mutation. So when this happens, the translation happens in a different way, and a canonical amino acid is incorporated in the structure. So that shows that this is not because of the transcription and translation process, but mostly the replication process, so that they lose their, they mutate into something else. So finally, they decoded 3 unnatural codons, but not 3 unnatural amino acids or non-canonical amino acids, because they didn't have like another technique like Spec or QAAC. So they used 3 non-canonical amino acids, and for the 200 codon, they used serine, which is probably a mutation in the 200 part. So when they did the mass spec, they got even higher results than they did before. So that also shows that location of the codon is also important. So I'm almost done. So I want to make like a small discussion. In this article, they created a first organism with 67 sense codons, excluding the 3 ones that are stop codons. They coded both canonical and non-canonical amino acids simultaneously. And what other thing they accomplished is that they showed that it's possible with 6 different base pairs, they can expand the sense codons or the amino acid cycle up to 213, excluding the 3 stop codons. So they created an extremely cheap method to produce proteins with non-canonical amino acids, because normally what they do is they manipulate this one of the stop codons, and they amino acylate the tRNA of that codon with a non-canonical amino acid. But in this case, we don't even worry about playing or changing the structure of the natural system, but we add to the natural system. So that makes it quite a bit cheaper than what the other studies has been done. And also, this has a really interesting drug delivery option, because some of the drugs, or maybe even the drugs as created as non-canonical amino acids, can be really helpful in terms of targeting drugs, such as if there's a drug that should be targeted in the endoplasmic reticulum, in a system with a signal peptide that translocates the peptide into the endoplasmic reticulum, that amino acid can be in that protein, and once that happens, we will just deliver our drug to the endoplasmic reticulum. So that's really nice, I think. So thanks for watching, and if you have any questions, I'm glad to answer them. So Cengizhan? I'm here. I think Özgür disconnected because he had like a connection error. So can we go on Turkish or English, depending on... Not an issue. Okay. So this article is published this year in April, and this topic is not really well researched area, since it's hard to work on this matter, since it takes a lot of resources and a lot of time to work on. So even this group that they're working on is working in a really good facility with immense amount of resources and money, probably. So it's understandable why there's not many studies done, and this is like the first time they actually did that. So in 2017, they created a semisynthetic organism that does the same thing, but with only one, but only adding one amino acid. So in this study, they wanted to show like they can also add three, at least three. So there's not any study has been done by using this system, but I remember there's some studies done on drug delivery by adding non-canonical amino acids into the proteins. I also remember one of our faculty members, especially Cem Hoca, Cem Albayrak, also mentioned about this in the presentation. So we can also check his studies and see if we got a something that we can do about it. But this way of incorporating a non-canonical amino acid is the first time. So it's possible and it's already been studied a bit, but I can't tell much about how they actually can use this system, this specific system that I just talked about in the drug delivery systems. My opinion leads on that many people don't really understand this specific way of delivering drugs, because there are like many other things like using viruses or using natural proteins. And also when people write grants, when they ask money, or when they want to do research, many people don't actually really understand how this system works. So that's one of the biggest factors why people are not really working on this matter. And another thing is, this sort of studies are really expensive, since they depend on really advanced biochemical reactions to produce these non-canonical amino acids. So I would say people still need to understand it. So the most important thing should be that people make other people excited and make them want to study this topic, so people can understand more easily and scientists can get more money for these studies, I would say. Yeah. I remember he mentioned that he has some biochemists in his group, or he has some people that he knows about biochemistry. So he's mostly working on synthesizing these specific non-canonical amino acids by dealing with these biochemical reactions. And there's some people in Koç University that can also do that. So he's mostly dependent on people who do biochemistry, and the rest is just way simpler than just creating the non-canonical amino acids. And about money options, I'm not really sure how he manages to go through with his research. Um, I'm not really familiar with his research for most part, because I'm mostly focused when I'm preparing this sort of presentation. I'm mostly focused on the genetics part of this topic. But I know there are many ways to develop proteins with specific functions, and as I mentioned before, they have uncommon bioorthogonal chemical groups. So they have immense possibility of options of creating affinity for something, or making something more catalytic, or making something more thermostable. So from that perspective, there's a huge way of using these things, since they can be implemented into any structure, and unlike the natural systems, these non-canonical amino acids can do things way simpler than the natural ones do, because in chemistry we can just do specific things such as creating affinity for something like this. I mean, maybe this azide could be replicated or faked, I would say, by using maybe 3 arginine groups. But in this thing, we can fake it by only using this amino acid. So that makes it very efficient for the studies, and every study is something new. Like even studies by just changing a single amino acid and analyzing the protein after this mutation is publishable. So by adding something completely different to the structure is publishable maybe multiple times, I would say. And about Jeff Hoja, I'm not really sure about his research. So is it a question? Yeah. So do we have any other questions on this matter? So maybe Özgür, do you have any question, or Buket?
No, not right now. So maybe
Okay.
I will. Yeah, I have some internet problems, just like Musra. Yeah, that's why. And then maybe I just read a bit more, and then I can just reach to you, and you can like answer me.
Okay. So for now, I think it's enough for the journal club presentations. If